Okay, SBI4U, this is the second video in homeostasis, and here we're going to talk about the brain and brain functions. Specifically, we're going to talk about this stuff. So we're going to do an overview of neuron anatomy first, then we'll talk about how neurons actually talk to each other, then neurotransmitters, very important chemicals, and a quick look at a reflex arc. That's the one that we saw in class, the knee-jerk reflex, the patellar reflex. Then we're going to move over to a different PowerPoint and talk about brain and nervous system structure, and then the divisions of the peripheral nervous system. And that's all we're going to get through today. This part of the curriculum just asks you to understand the physiology and function of the nervous system. So we're going to start with the basic unit of the nervous system, which is, of course, a neuron. So let's start talking about that. Here's a neuron. Here's the physiology of a neuron. And here's showing you the information flow. What's important here is we've got the cell body, also called the soma, S-O-M-A, like somatic, the same word. So soma for cell body. It has two types of projections from it. The first are called dendrites. Dendrites from, I believe, the Latin for tree, because they look like little trees. And they bring in information from other neurons or from other places. They bring in information. And then this thing is called the axon, or the word the axon. It's very, very long, and it branches out at the end. And it passes it off to either more dendrites over here. So, this might project to the dendrites of a different cell, or it'll attach to a muscle or something, something like that. Maybe a gland to make it release something. These things attach to all kinds of stuff. A lot of them do attach to other dendrites, though. So dendrites, soma, axon. That's the information flow. Dendrites, soma, axon. The soma has all of the cell stuff in it. So, the nucleus and all of the protein-making stuff. So this creates the neurotransmitters and things, and then different transport molecules will push it along into the axon and into the dendrites and spread it around the cell. So that's a basic neuron. We were interested in how they talk to each other, so the nerve impulse or the action potential. It's done as the movement of ions, because they're charged, right? Ions are charged particles, and it goes between the membrane. It's actually nice that we do metabolism before we do this one, because you can understand the idea of an electrochemical gradient now. We had an electrochemical gradient in our mitochondria that was generating ATP for us. Here we're going to see electrochemical gradients, but what they're going to do is we're using the electrical part of the electrochemical gradient to send a signal. So those are going to send a signal. So here's one of the most important things about a neuron is its ion channels. They have channels for specific ions. They have channels for sodium, potassium, and chlorine, and it's a very interesting thing. These channels can open and close in response to different stimuli. Remember, different proteins will react to different electrical environments. So some of these channels will open up in response to electrical activity that's happening further away, and other ones will open up due to other electrical activity. Some of them will open up because of heat, or because of mechanical stress on the membrane, or because there's a certain amount of sugar in your blood, or really a lot of different things. So this is how your cells can recognize, your nerve cells can recognize that something has changed in their environment. There are different signaling molecules, different channels there. There's a resting potential in the membrane. Remember how in the mitochondria there's a bunch of H plus in the intramembrane space and little H plus in the matrix? The same thing happens here. You have a positive charge on the outside and a negative charge on the inside. This is something called a polarized membrane, and the millivolts, 70 millivolts, volts is a measure of potential. You can think of it as how much energy a positive particle would release falling through the gradient. You could theoretically measure the mitochondrial membrane's potential in volts as well, or millivolts, I suppose, because there's an electric potential built up there as well. Why is there a difference? The difference happens because of potassium and because of other things, potassium, sodium, and negatively charged proteins. There's a lot of potassium inside the cell compared to the outside, and a lot of sodium outside the cell compared to inside. And there's a lot of sodium, so the outside becomes more positive. You might be wondering why the potassium is on the inside if we're trying to make the outside positive and the inside's negative. Well, we'll get to that in a couple of slides here. Those are these negatively charged proteins inside the cell. So here's a picture of it. You see lots of sodium and lots of chlorine on the outside. You can kind of easily remember this by thinking about it being salty on the outside, right? Sodium chloride is the main ingredient in salt. So salty on the outside. And the potassium, it's OK on the inside. It's K on the inside. So potassium on the inside. And there's these negatively charged proteins and phosphate groups that are helping it stay relatively negative on the inside. So that's the setup of the cell, the nerve cell, the neuron. The inside of it, the cytosol, the cytoplasm we call it sometimes, is negatively charged. The outside of it is positively charged. So what's going to want to go where? Well, the sodium is going to want to flow towards the negatives, and the chlorine is going to want to flow towards the positives. The potassium is already with the negatives, so it's pretty happy right now, but we'll see what happens to that later. Why is there so much potassium on the inside, and why is there so much sodium on the outside? There's a pump. So it's called the sodium-potassium pump. It moves sodium out and potassium in. But the potassium can always move kind of by itself. And the sodium on the outside makes it be positive, so the potassium floods in. So you see that there. And here's the pump. Here's the sodium-potassium pump. My face is up in the corner here. I'm just going to hide the webcam for a sec while I show you. Well, when I get to this slide, I'll show you the thing. Actually, there's nothing up here. Here, I'll just show you that there's nothing up there. See, there's nothing up there. So, okay. Here's how it works. 3 sodiums come in and an ATP comes in. The ATP drops off its phosphate. Remember that releases a burst of kinetic energy because we're reducing the potential energy here. So kinetic energy gets released. That change lets the pump flip from down to up. Suddenly it flips from down to up. That kinetic energy kicks it to the up configuration. And then potassium comes in. Potassium comes in and sits on these two sides. And the potassium comes through. It doesn't need energy to come through because of the negative charge under the membrane. Inside the membrane is negative, so the potassium wants to be there, so there's no energy needed to come through. So every 1 ATP, this thing pushes 3 sodiums out and 2 potassiums into the membrane. This is in neurons required to restore this gradient after they fire because the firing is going to upset the gradient. But all cells have these, not just nerve cells, and I've written Nerev cells here. I'll fix that in the real PowerPoint. But all cells do actually have these. They're used to manage water balance because where the salt goes, the water follows. So you can turn on these pumps and change how much ion is on one side or the other side of the membrane because it's 3 out for 2 in. So your cells can then manage how much water goes in and out of them. It's a very important thing for a cell to do. They can manage that. It also helps facilitate diffusion processes, especially the sodium being on the outside of your cells. You can have the sodium be required for certain molecules to come through the cell. It's like a form of kind of active transport, but not really. It's called facilitated diffusion. Facilitated diffusion. So all your cells have these pumps, and they use a lot of energy. So if I just open this up down here, it uses up to 30% of a typical cell's ATP. Just your normal everyday cells, 30% of their ATP energy is spent shoving sodium out and potassium in because they use that gradient for all kinds of interesting cell processes. But in neurons, it is the main way that they fire. That charge is the main way that they fire, and they want to maintain that gradient all the time. Up to 75% of your neuron's energy goes to this one little molecule. So all told, it is about 35, maybe 1/3 of your energy, about 35% of it, of all of the energy in your body is just going to pumping sodium ions out of your cell and pumping potassium ions into your cell, which I always found kind of cool. Now we're going to talk about the action potential, so I'm going to pause. And we're back off pause. We're going to talk about the action potential. This is called the action potential. What you're missing up here is just potential, which is, that word down here. How this works is the neuron gets stimulated. So it gets some kind of signal from the other neuron, or heat will activate a channel protein, or maybe some twisting of the membrane will activate a channel protein. That will let a little bit of sodium into the cell. And if it lets enough sodium into the cell, it might reach something called threshold. And at that point, a cascade of events happens that makes the whole neuron fire. So here's what you're seeing there is a stimulus. The stimulus at -70, the stimulus will open up little channels and let a little bit of sodium in, if there's not enough, the pumps will just reset it back to the resting state, right? But if you get enough stimulus in a short enough time, you end up opening those ions, and you get this big spike. The spike actually goes past 0. Here's 0 here. The spike actually goes past that. And then the pumps will return it back to normal. So that's the big spike. Here's kind of the way that it works. The sodium ions, when those voltage gated channels open, the sodium ions will suddenly rush through, and they're attracted to the negative on the inside. They rush through so fast that it actually goes past negative and goes to the positive because of the potassium that's inside. So once it's in the positive, the sodium channels close. Then at that point, when it's negative to positive, it's depolarized, it's actually flipped. Now it's positive on the inside and negative on the outside. Guess what's going to happen to the potassium ions now that it's positive on the inside and negative on the outside? Well, they're going to flow out. Right? The potassium is going to flow out. Right? Potassium ion channels open up, potassium rushes out. It goes back to negative on the inside and positive on the outside. This is called repolarization, but it does actually go past. So the potassium will help it repolarize, but it does go past that, which we call hyperpolarization. During that time, it's very hard to make the neuron fire again. This whole process, all of these things opening and closing, happens in like a very, very brief time, 6 milliseconds. And on average, your neurons across your entire body and brain, they fire approximately once a second. Like each neuron fires about once a second. So it goes fire, fire, fire. Much faster for neurons in your eyes and the neurons that are in your visual field. Those can fire up to 250 times a second, so they can fire very, very quickly. And even faster rates have been reported. But yeah, they can fire very, very, very quickly and reset themselves very, very, very quickly. Yeah, there's a car alarm going off in the background. That's great. This is an all or nothing principle. If it's strong enough at one point to set off the action potential, the whole neuron is going to fire in sequence. And the best way to explain that is over on the whiteboard. So I'm going to pause and go over to the whiteboard and kind of explain this stuff. Okay, so I'm over here on the whiteboard, and I've drawn a little diagram of, this is a normal neuron at rest. And we've got 3 channels. We've got the stimulus-gated sodium channel. We've got the voltage-gated sodium channel, the voltage-gated potassium channel, and another voltage-gated sodium channel. So it's the same as this guy. And right now they're all closed. All of them are closed. So there's a little cap on here, a little cap on here, a little cap on here, a little cap on here. This isn't what they actually look like. It's just kind of an example. What happens when your nerve cell gets a stimulus, there's the protein here, the stimulus-gated channel, responds to heat or sugar concentration or something, right? And it opens. The lid comes off for a second. When the lid comes off, sodium can flow through. Sodium can flow through. And you see the sodium can make it through. Sodium starts going through the channel. If the stimulus goes away, the lid comes back on, and nothing happens. But if enough sodium comes through that it goes from -70 to -55, right, -55 millivolts, if enough sodium comes through, then this guy opens up. At -55 millivolts, this guy opens up. And all of a sudden, a lot of sodium can start coming through, independent of the fact that there was stimulus over here. You could take away whatever made this go, and this is still open, still sending sodium through. So the sodium starts coming through. Right, the sodium starts coming through. That happens so much that in this area, the sodium all leaves. Right, all the sodium actually manages to make it all the way through. Right, all the sodium manages to make it all the way through. And because all the sodium is now gone, what's left is the chlorine. Right, the chlorine minus, and it's not making, it can't make it, but it can't make it through here, and this thing shuts. This thing shuts, but now it's minuses, right? Now it's negative around here. Negative in this area, just in this area, because all the sodium is gone, all the sodium is down here, and it's more positive down here where all the sodium went. So this is now suddenly more positive, and it's a full inversion. It's polarized the wrong way. Right. Negative and a positive here. That prompts this channel to shut. Stops the sodium from coming back the other way right now. And the other thing that happens, this is the really important thing, this is why the signal can propagate, is now that there's a negative over here, there is a pulling force on this side for all of these positive molecules to kind of start running away, right? To start going this way, because there's negatives here and the positives here. And here, these negatives are going to get attracted. These negatives are going to get attracted to this positive area down here. And wouldn't you know it, that's exactly what this channel needs to hit -55 millivolts, because it's seeing less positives on top and less negatives on the bottom. It's getting less of that potential, that buildup of difference in charge. So it then opens. It suddenly opens. Right? And the sodium that's over here can rush through. And now this part of the membrane depolarizes, turns negative on top and positive on the bottom, and then, wouldn't you know it, there's another one, right, over here. There's another one of these sodium channels over here, and then another one, and then another one, and it just goes on and on and on and on down the line. The signal won't propagate backwards because of the hyperpolarization that happens. The potassium channel will open up, and the potassium comes back through. The potassium comes back through and equalizes everything again, and you go back to your start. But that's the important thing, is these voltage-gated sodium channels are what passes along the signal. And then, of course, the ATP has to come in and get the sodium back over here and get the potassium back over there. That's an action potential in a nutshell. So here's where we're at in the note because of that, the all or nothing principle. Because it's all done based on a chain of voltage-gated channels, as soon as one of them goes off, they're all perfectly spaced apart so that that signal just goes on down all the way to the end of the nerve cell. Now the next piece is what happens when it reaches the end of the nerve cell, and that's neurotransmitters and neurotransmission between two neurons. This happens at something called a synapse. A synapse, it's small spaces between the neurons. So the signal gets to the end, and the neuron doesn't touch the next neuron, because if it was touching, then the signal will just continue on right through without any chance to modify it, and our brain wants to be able to modify the signal. We don't want to react in the same way every single time. We want to be able to change it. So what happens is you've got this gap, this little tiny gap, and that's called a synapse. And the gap is between the axon of the signal coming in and the dendrite of the next neuron. This way your brain can tell the neurons to kind of ignore or pay more attention to each other's messages. And it does all of that through neurotransmitters. Here's a picture of one. All right, here's the axon of this one, the purple axon. Just barely staying away from the cell body, from the soma of the next cell. Yeah, so that's the synapse right in here. Synapse right in there. At the end, it reaches the what's called the axon terminal. Sometimes they're called terminal buttons because they kind of look like buttons, little buttons, or boutons, we use the French, bouton. They release vesicles, these little vesicles that have neurotransmitters in them. They travel across the cleft very, very quickly. They diffuse across the cleft very, very, very quickly. To give you an example of how quickly they get across that cleft, even your reflex actions like touching a hot stove and pulling your finger away, that requires at least one step of synaptic neurotransmission. Actually, I think it requires at least two. Yeah, they bind to receptors on the other side, and that creates some kind of response in the other neuron. It can make it more likely to fire or less likely to fire, depending on which neurotransmitter it is. The more likely to fire ones are called excitatory. There's the word up there, excitatory, and they'll make it more likely to fire. But that next neuron might be inhibiting something else, and so it's like it's almost like a little computer in there that has code. It's really interesting. So that's another picture of it. One thing to note is that the release is actually caused by calcium channels. So calcium ions, specifically voltage-gated calcium ion channels, they flood calcium into the bouton, and that calcium input sets off a chain of events that causes these to send out their neurotransmitters. So yeah, that's that. And then there's the receptor, and the receptor will either make the next neuron more likely to fire or less likely by messing with sodium, potassium, and chlorine levels in the neuron, in this next neuron. There's a name for these two. This is called the presynaptic neuron because it's before the synapse. It's sending the signal. The one that's receiving the signal, which is after the synapse, is called the postsynaptic neuron. And here's the neurotransmitters themselves. I'm going to move my thing down in the bottom corner here. The first one, and probably the most important one to know, is glutamate. Glutamate is the all-purpose neuron activator. It is your primary excitatory neurotransmitter. If the presynaptic neuron sends glutamate out into the synapse, the postsynaptic neuron will be more likely to fire. Maybe it'll open up some sodium channels, and it's already got some sodium channels open when the signal comes along. It makes it more likely to fire. GABA, on the other hand, gamma aminobutyric acid, is very, very highly distributed in the brain. It's your all-purpose inhibitory neurotransmitter. So it inhibits the next neuron. It makes it harder for the next neuron to fire. And so the balance of glutamate signals to GABA signals acting on the neuron is kind of what decides whether it's going to fire off or not. Whether it's going to fire or not. Just a little aside, alcohol acts on GABA. It's the primary thing that alcohol acts on. Alcohol increases the effect of GABA. It increases the inhibition. You might have heard that alcohol reduces inhibition. Well, it reduces like social inhibition kind of stuff, and that's a high order brain function. It requires a lot of connections in your brain thinking about people's opinions and stuff like that. if you increase the effect of GABA, it slows down that part of the brain, and that part of the brain just kind of goes, maybe not. It doesn't really feel like working on it. Another one that's important, and I'm going to move this back over, is acetylcholine. It is the one that is released to activate muscles. So when your nerves touch your muscles, they release this acetylcholine stuff, and then the muscles know that they have to contract. A few more. Epinephrine and norepinephrine. You might have heard of them as adrenaline and noradrenaline. They're very, very similar. Norepinephrine does more brain stuff, and epinephrine does more body stuff. That's why epinephrine is the stuff in the EpiPens. The epi stands for epinephrine. The norepinephrine in the brain, when it releases, it increases your attention and your memory, especially in stressful situations. It makes you pay attention to tasks a lot more. So, when you're panicked at, 10 p.m. trying to write a paper that's due at 11 p.m., your norepinephrine's all high. Epinephrine, on the other hand, does more of the body stuff. It'll raise your heart rate. It'll make you breathe faster. It's involved in that fight or flight response. Then the last two, very heavily implicated in depression and addiction and stuff like that, so they're important to talk about. Dopamine is your reward chemical. Your brain releases this in all kinds of situations when it wants you to do that thing again. So that's dopamine. It can be excitatory in some parts and inhibitory in others because it has this complex role, this reward role. And a lot of drugs do act on it. So a lot of illicit drugs will directly stimulate dopamine receptors. This, for example, is the difference between amphetamine and methamphetamine. Methamphetamine has a much higher affinity for dopamine, and it turns into an addiction much faster than just normal amphetamine, which is like the stuff in Ritalin and other ADHD type medications. The other one is serotonin. Serotonin is very, very heavily implicated in depression. It is generally excitatory, but again, it can excite an inhibitory thing. It regulates your mood. It is an anxiety. It's implicated in anxiety. It also does work in the digestive system, the immune system, and in nausea, which of course is linked to the digestive system. So this is why, depressed people with depression will often have issues with, food, and they'll have immune problems as well. There's a whole splash of associated symptoms. You might have heard of chemical imbalances in the brain, and that different drugs and therapies are there to address that chemical imbalance. Well, these are the imbalances that they're talking about. It's imbalances in the actions of these neurotransmitters. Some people's brains are just more sensitive to dopamine or more sensitive to serotonin, or specific regions are more sensitive or less sensitive. And that's the kind of stuff that neurologists and psychiatrists are trying to fix. We're going to get into the last part of this note, which is a simple example of neurotransmitters and a look at a reflex arc. I'm not going to change I'm going to change this down here or this up here. I'm just going to stick around, keep it around. This is a reflex arc. What you're seeing here is a stretch receptor. The orange is a neuron, a stretch receptor. It's going to the spinal cord. This is the spinal cord here. And the spinal cord is doing a little bit of processing and then sending a signal right back to the muscle. So this red is the muscle signal. It's actually sending two signals, one to the same muscle that the stretch receptor is on, and one to the opposing muscle, the muscle on the other side of the leg. So what happens here is the stretch receptor feels a stretch. The deformation, the actual twisting of the membrane, of the cell membrane, sets off an action potential. Sodium ions flow in, and then more sodium ions and more sodium ions, and that travels all the way up to your spinal cord from basically your thigh. So think about how far that has to go, and it's a single neuron. It's one single cell that's that long. It gets to here where the axon of the neuron, right, goes to the cell body and then continues on to the axon. That axon is interfacing with two different neurons, but both of them get the same signal. They get a glutamate signal, This one, the glutamate signal to the red here, that one, goes straight back to the muscle, and releases acetylcholine, which makes the muscle contract, which tries to stretch the leg. The other one goes to this interneuron, so it's a very short neuron, this little green neuron, and it's still a glutamate. It excites the interneuron, but that interneuron releases glycine, just the amino acid, which actually acts as a neurotransmitter on its own, and that glycine will inhibit this motor neuron. So this same excitatory signal from the same neuron had both an excitatory effect on this muscle, but an inhibitory effect on the other muscle, thanks to the interneuron in between that turns the excitatory signal into an inhibitory signal. It really is a lot like computer programming. You have like loops and things. Really, really interesting. Both of these will control muscles by releasing acetylcholine. This one, because it's more stimulated because of the glutamate it got, will release more acetylcholine. This one, because it's inhibited because of the glycine that it got, will release less. And the total, the sum of that is that the muscle, the leg, will rise. And this is called the stretch reflex because it lets us keep position. So like if I put my arm like this, I've got muscles here and here holding my arm in place. And the stretch reflex, when my arm naturally moves up a little bit, will pull it back down. When it moves down a little bit, it'll pull it back up. And of course, there's all kinds of other neurons attached to these muscles, and they're putting out their own signals. So there's conscious control muscles that mean that I can push my arm up and down and up and down. But if I want it to just go in one place, I don't need to really think about that. It just is taken care of by my spinal cord in this stretch reflex. This very, very simple stretch reflex. But even just this simple reflex requires glutamate, glycine, and acetylcholine, 3 different neurotransmitters for 3 different synapses. The last piece here is just a look at some different motor neurons. Here's what a motor neuron looks like. Right, it's sending it right down to the motor, the muscle cells. So just sending signal right to the muscle cells. This is a sensory neuron where it just has a receptor cell on one side. It's not connected to any neurons because it's trying to connect. And it sends the signal out to different neurons. So for example, this one here sends 2 signals, right? The 2 branches to 2 different things with the same neurotransmitter. And then here's an interneuron which takes in signals on one side and puts out signals on the other side. So that's this little green guy here. And that's basic neuron anatomy and how the brain and the nerves make decisions and take actions. So this is a really good example of that. I'm going to pause and go over to the brain note. It's a lot shorter, so I'm going to pause. Okay, so for the last part of the lesson, we're talking about brain anatomy and nervous system anatomy in general, just the general scheme of the nervous system. And we're going to talk about divisions in the nervous system. So there's the central nervous system and the peripheral nervous system. That's the first big division. I'm going to make a chart of this at the end of this lesson. So don't worry, we're going to have a chart. Central nervous system is central. It's your brain and your spinal cord. That's it. It's the main control center in terms of homeostasis. Then there's the peripheral meaning to the outside. Think about like perimeter of a shape, peripheral. They're connected to muscles and glands and organs, various receptors for feedback. So that's going to pull in information to the central nervous system so that your brain knows what's going on, and then send out the decisions that the brain makes. So that's the central and peripheral. Peripheral is made up of what we call nerves, which are actually bundles of individual neurons. So a nerve is a bundle of neurons. Some general brain structure here. General structure of the brain, we have, I'm going to move my thing over. We have the brain. It's protected in the skull by a few layers of tissue. The skull is bone. Then there's the dura mater, which means hard matter. Then the pia mater and the arachnoid layer. So it's almost like a little tiny crash helmet in there to help your brain handle the little bumps that it gets now and again. It's not particularly strong, but it's strong enough for the basic everyday stuff. If you hit your head too hard, though, you end up with basically a brain bruise, which we call a concussion. Yeah, or more of a brain cramp, really. So this is the blood-brain barrier. There's also blood going all through the brain, but this blood-brain barrier is there. So the blood vessels in the brain have a special barrier around them that only lets certain things through. Otherwise, bacteria and viruses and stuff could get into the brain, and that's just a bad time. One of the things that can happen when a bacteria gets in there is meningitis, and meningitis just means a swelling of the meninges, and that's these 3 layers here. And here you see some blood vessels traveling around the brain. Then there's the spinal cord. I'm gonna put my my thing back in the corner. Spinal cord carries the messages down from the brain and brings the sensations up from the brain. The dorsal nerve tract brings the sensory information up, and the ventral nerve tract carries the motor information down, all of the conscious motor control down. So if you know enough French, dorsal comes from dos, means back, and ventral comes from ventre, which means stomach. So the back side of the spinal cord is going to bring sensory information up, and the stomach part, the front part of it, sends it down. So here's a picture of it with the two sides, the ventral root and the dorsal root. And here's the front and the back for the spinal cord. This is how this is why epidurals come in through the back. Epidurals, when women are giving birth, they're trying to freeze the dorsal tract, which is bringing the sensory information, but they don't want to affect the ventral tract. They want to make sure that the woman can still push and stuff, but they don't want her to feel the pain information coming up the back. So that's the idea. Here's some detailed brain structure. We've got the forebrain, midbrain, and hindbrain. I realize it's more like top brain, middle brain, and bottom brain, but this comes from rat studies on rats and other animals, and rats and other animals are, they face forward like this. So for them, they do have a forebrain, a midbrain, and a hindbrain. But for us, who, we're mostly upright, the forebrain is at the top, the midbrain's in the middle, and the hindbrain's at the back. The forebrain has 3 different structures in it: the cerebrum, which is all the thinky part, the thalamus and the hypothalamus. The thalamus and the hypothalamus have to do with regulations and emotions, and kind of integrating stuff. So this makes decisions. This kind of modulates the decisions that are made based on emotions and other responses. Then the thalamus and the hypothalamus can affect the rest of the body through hormones. They're linked to the pituitary. Here's the pituitary gland. It actually hangs outside of the brain. I don't have a picture of it. We'll see that picture in the endocrine unit when we talk about hormones. But there's brain, and then this little guy hangs out into the blood, so they can send nerve signals to the pituitary, and then the pituitary will send out signals to the body to take action. So yeah, so for example, if the hypothalamus realizes that you need to raise your body temperature, it will send a signal to the pituitary to send out hormones that will raise body temperature throughout the body. The midbrain is less developed. It's sometimes called your emotional center. Really the very, very basic emotions: fear, hunger, that kind of thing. It does act as a relay center for some of the eye and ear stuff, because the eyes are right in front of it and the ears are right next to it. But it's also where the dopamine, all the dopamine in the brain is basically made here and projects from here. So reward and addiction actually occurs below the level of the brain that's responsible for conscious thought. people with physiological addictions, like actual addictions that have affected the brain structure, they really aren't consciously in control of that addiction mechanism. They can exert conscious control over it, but it is a deep brain problem rather than a just think about it and it goes away kind of problem. Then there's the hindbrain, which is also called the reptilian brain, because even reptiles have one. Cerebellum, pons, medulla. This is all of your automatic stuff. So pons and medulla, the brainstem here deals with the automatic regulation of breathing, the automatic regulation of heart rate, and things like that. The cerebellum is really cool. It looks like a little brain. That's what the name is. It's cerebellum. It actually has half of your neurons. Half of the neurons in the entire brain are here in the cerebellum. It's involved in fine motor coordination. So the ability to really, really finely coordinate your muscles and do stuff like writing and things like that are in the cerebellum. And it needs a lot of neurons very, very closely attached to each other in order to make those fine coordination events possible. I'm going to link a video down below that is absolutely adorable. It's dogs and cats that have what's called cerebellar hypoplasia, so they just had some kind of issue when they were developing and this didn't develop enough. And so they still have control of their bodies, they still know what they want to do, but they just can't get their coordination quite right, and so they're really clumsy. And it doesn't hurt them or anything like that, they're just kind of moving really weird. So it's really, really cute. I encourage you to watch the video on cerebellar hypoplasia down below. Hypoplasia down below. Okay, next up we're going to talk about divisions of the peripheral nervous system, and that's everything else that isn't brain and spinal cord, so we're going to pause for a sec. Okay, so the last piece here is the divisions of the peripheral nervous system, and they only apply to the peripheral nervous system. So peripheral is everything that's outside. It's going to send signals from the brain out, and it's going to bring signals from the body back to the brain. And that makes our first division, the afferent system and the efferent system. Think about efferent like exit for efferent, or effort, right? Effort, it almost looks like effort. Or effect. It sends the signals out. Oh, my fish tank light stopped, so there's less light in here. I'm going to turn on this light. The efferent system. Efferent system carries it away. Afferent system brings it in. Effect, affect. So afferent is in, efferent is out. Yeah, you can think about it maybe as like A comes before E, so the signal into the brain comes before the brain's decision for what to do with that signal. That's another way that people think about it. There is a further subdivision here in the efferent system, in the signals going out to the body, and the division is based on whether we're consciously aware of it or unconscious, or we're not aware of it at all. So somatic is the conscious control, and autonomic is the unconscious control, because it's automatic. So think about automatic. Soma comes from the word for body, so somatic means, I guess, body, things that can move the body, skeletal muscles mostly. That's stuff you can consciously control. Meanwhile, autonomic would be stuff like making your intestines move, right? Making your heart beat. You can't force your heart to start beating. You can't just tell your muscles to beat. The autonomic has to take care of that part. So yeah, somatic, conscious, voluntary goes down the spinal cord. So it goes straight down the spinal cord. Autonomic kind of regulates things from different places. But there's two major divisions in that one too. There's the sympathetic division and the parasympathetic division. Sympathetic will speed things up. Sympathetic speeds things up. Parasympathetic slows things down. So sympathetic speeds things up. Parasympathetic slows things down. And that's only an autonomic nervous system feature. It's interesting that they come out in different places in the spinal cord. The parasympathetic, the slow down signals come out higher up, and the sympathetic, the speed up signals, come out further down. And there's some parasympathetic ones lower as well. Good example of this is when you have a stressful situation, your sympathetic autonomous nervous system helps you prepare for that. The full name of this would be the sympathetic autonomous peripheral nervous system, because it's going out to the organs and stuff like that. It's not just staying in the spinal cord, and it increases your heart rate, it dilates your pupils, it redirects blood to your muscles, makes the blood go faster to the muscles. How do these help? Well, heart rate means you have more oxygen circulating. Pupils dilated means you can see better. And blood to the skeletal muscles means your muscles are ready for action, ready to use those energy molecules. And there's the location, right? The sympathetic is right in the middle of the spine, and the parasympathetic is top and bottom of the spine. Just kind of an interesting thing. And that's it. So let's look at what we talked about. We talked about the neuron anatomy, where it comes in on the dendrites, moves through the soma, and then goes out to the axon. We talked about the action potential and the different ions that are moving and how that works. We talked about neurotransmitters and how they work in a reflex arc. And then we went over very briefly basic brain and nervous system structure and the divisions of the nervous system. So I'm going to go over the divisions of the nervous system just over on the whiteboard, and then we're going to be done the lesson. So just a second. Okay, I'm back over here. And we are going to just do this very quickly. We have the first division was central versus peripheral. Central versus peripheral. Central is just brain and spinal cord. The brain can be forebrain, midbrain, and hindbrain, but we're not worried about that. The peripheral can be further subdivided into two things. The peripheral can be subdivided into the afferent, which is the senses, and the efferent, which is the control. Right, is the control. And the efferent can be further subdivided into somatic and autonomous. Right, somatic means conscious control, mostly just skeletal muscles, and autonomous is unconscious. And those unconscious signals can be excitatory or inhibitory, right? They can speed up all of those automatic things like heart rate, or they can slow down all of those things like heart rate. And that's the last division here into sympathetic and parasympathetic. So you got that, parasympathetic. So this is sympathetic is up, right? It makes things more likely. Parasympathetic is down, makes things less likely. So sympathetic works you up, parasympathetic tones you down. And that's the end of the lesson. Thank you for paying attention, and I'll see you guys in class.